Our next speaker is Madeline Lancaster. Madeline completed her PhD in 2010 at University of California, San Diego. She then joined the IMBA in Vienna for her postdoc research. And in 2015, she established her own lab in the cell biology division at the LMB. Welcome, Madeline.
So I have been at the LMB for 7 years now. I've had my lab since 2015. So I'm American, as you can tell. So I'm sort of one of these good examples that we've been hearing so much about of this sort of cross-contamination across the Atlantic. Yeah, so I thought I would spend this time just talking about some of the work we've been doing since I've started my lab at the LMB and highlighting the collaborative work that's been made possible by this really unique environment. So we've heard a lot about molecules, we've heard a lot about subcellular compartments and cells themselves. Sarah's talk is starting to get a little bit above the cell now into multicellular kind of things. I'm going to talk about tissues now. So I'm in the cell biology division, but we work on the brain. I'm not in neurobiology, but I will also show you some interesting parts of the brain that don't involve neurons that you've probably not even heard anything about. But ultimately, really what we're interested in is tissue development, and we're sort of focusing on the brain. And so we're interested in how this complex multicellular tissue forms. And in particular, we're interested in human-specific processes that really set us apart. Because, I think most of us would agree that our brains are really one of the key differences about humans compared with even our closest living relatives like chimpanzees and gorillas. And so I'm not going to have time to talk about some of our evolutionary work, but I'd be happy to talk to anybody about it if you want, because I can go on and on about it. But we are interested in really what are the mechanisms that are really unique to humans. And when I say really unique to humans, I mean different from, for example, chimpanzees and gorillas. So because we're interested in such a human-specific question, this is very difficult to look at in sort of standard animal models. So we've learned a lot from mice and fish, and we've about the basic developmental processes that lead to early, that are responsible for the early stages of brain development, but we don't really understand that much about why our brains are so enlarged, for example, or what leads to this beautiful connectome. And so I thought I would put this up first, just because I think it's fitting. And so because we're interested in these human-specific questions, we need new tools. And, as Sydney said, progress in science depends on new techniques, new discoveries, and new ideas, probably in that order. And I would wholeheartedly agree with that. And so in my lab, we kind of have two sort of strands we're going in. One where we're doing a lot of method development so that we can make these new human models that will give us the ability to ask these human-specific questions. And then obviously using those to make new discoveries. So, so what do? We make organoids. So we call these cerebral organoids or brain organoids. And what these are basically an in vitro model of the early developing brain. So I'm showing here initially just sort of a diagram of the key stages of early development that we first need to model in a dish before we can even start getting to something that looks like a brain. So you start with the early embryo, the blastocyst, and the inner cell mass is what will give rise to the embryo. It will undergo gastrulation, and then the neuroectoderm here will fold up and close and form the neural tube. And then, of course, the more anterior portion of that is what will give rise to the brain. So how do we model this in a dish? Well, basically, we just let the cells do what they're already programmed to do. So embryonic stem cells are already ready to build an embryo, and we're essentially giving them the environment to do so. We're sort of just sort of the permissive environment without necessarily driving them in any way, really. And so this is work that I did during my postdoc when I was in Vienna. And so what we found was that it's really quite remarkable how robust this process is. These cells, if you simply take them off of their 2D environment and start culturing them as these little 3D balls, they'll start spontaneously trying to make the germ layers of an embryo. And so these are called embryoid bodies. And as I've told you, one of these germ layers, the ectoderm, and in particular the neuroectoderm, will form in particularly around the outside of these aggregates. And then when we place this ball of sort of a mix, there's some sort of mesoderm and endoderm on the inside and neuroectoderm around the outside. When we put that in a droplet of matrigel, which is just a protein-rich gel that sort of mimics the ECM, then this tissue will reform, sort of close up and form these neural tube-like buds, which then will expand and form little sort of brain tissues. And we've, of course, with the help of a little bit of agitation to help with nutrient and oxygen exchange. Since I started my lab here, though, we've been taking these methods further, and in fact, we're not even using the original protocols that we established when I was a postdoc. We've really come a long way, and we've now been able to make this happen in a much more robust fashion so that we get more reliable formation of neural tissue, and in particular, so that we also specifically get more of the cerebral cortical tissue that we're interested in. So this is a section of a human embryo, a sagittal section of a human embryonic brain. And basically, this is one of our organoids. I know it doesn't look anything like what you're seeing here. But actually, each of these, you see each of these little sort of buds growing off of here. In fact, each of these little buds is kind of like a little cerebral cortex growing off of this organoid. So if you don't believe me, I'll just try to show that by looking at some histology. Here's one of these organoids sectioned and stained for a couple of markers. In this sort of magenta color, you see neural stem cells, and in white are the neurons. Here is a mouse embryonic cortex during development. And so what you can immediately recognize is that each of these little buds that I was pointing to has these fluid-filled spaces, just like the developing brain. And you have this beautiful layering happening, which you can see a little bit better here, where we've zoomed in on a wall of one of these cortical tissues. And you can see the same kind of layering that you see in the actual developing brain, where you have progenitors on the inside, and then you have neurons that migrate out and form the cortical plate on the outside. And the cortical plate will then become the gray matter of the brain, and these cells, of course, will send their axons inside and form the white matter. So we've also been able to find new ways of getting better maturation of these organoids. So previously they were pretty good at making neurons, but then the neurons would kind of die because they just weren't really in the environment that they needed. And so we've discovered this way of basically doing slice culture on organoids, and we can get much better survival of neurons and very nice connections being made. So here you can see a section. So one of these slices where we've stained for axons and dendrites, and we get very mature neuronal morphology as well. So these slices can be kept for years, actually. And so, for example, these beautiful neurons with dendritic spines, these have been kept in culture for a year. And so we're getting really nice, mature neuronal morphology. And then we collaborated with Laura Mazzula in the Tripodi lab, because we're a developmental biology lab. We didn't know what to do with the neurons once we've made them. So we went over to neurobiology and asked them, can we actually measure these neurons and see if they're actually active? And she really helped with doing some patch recordings on these neurons. And then we started collaborating with our next door neighbor, Wanda Kukulski, who had started her lab at about the same time as me, actually, and was working on looking at ER contact sites, mainly in yeast, but also in HeLa cells, and was doing this by cryo-tomography. So we had this crazy idea that could we actually use these slice cultures that we'd established, which, like I said, make these beautiful axons and dendrites, and actually even make these long-range tracts that projects away from these slices. Could we actually then use these to start doing cryotomography on axons? Because the beautiful thing as well about these cultures is that because they form these tracts that leave the organoid, by putting a grid next to the organoid, you can get a pure preparation of just axons. So you can really know what does the inside of an axon look like? So we did cryo-clem with Wanda. And so this is a movie of, first we've done live imaging work because we've GFP labeled a subset of neurons within these slices. And then we can actually watch the axons grow onto the grid. You'll see that they like the grid bars in particular, which is a slight problem, but every now and then they go off the grid bars and we're able to see something. It's probably a good thing actually, because that means that in between the grid bars, we have a nice thin section. So then, and then you'll see the cryo light microscopy and a zoom in all the way into the EM. So here you can see, like I say, this is just a time lapse, just an image taken every day. And you can see how the axons are gradually getting there. Once we see them getting into the middle, we plunge freeze. Here's the cryo light microscopy. In purple, we've also stained for sirtubulin. So we can also just see all of the axons. Here's a nice little bundle of axons. Now we're zooming into one particular axon and zooming in all the way to the point where we can actually see a vesicle with its cargo, which I think is just so amazing. So what this, so I won't talk really much more about this other than just to say that now we have this really cool tool. We can start looking at the actual subcellular architecture of human axons and look at them at different stages, for example, and see how axon cytoarchitecture changes over development. Then I wanted to switch gears a little bit because this is another great project that's involved fantastic collaborations, and start talking about cerebrospinal fluid. So cerebrospinal fluid, many of you have hopefully have heard about that, is typically talked about in textbooks as just kind of being there to, I don't know, buffer the brain in case you hit your head, so so your brain doesn't hit the skull or something. But it actually does so much more than that. And I think its primary role really isn't that, but rather essentially turnover of nutrients and clearance of toxic byproducts. So there's a lot of new exciting research in this front. And I think it has a lot of implications also for neurodegenerative disease, for example, and just healthy brain aging. And so the CSF is produced by this tissue here on the inside of the ventricles called the choroid plexus. So this is that part of the brain that I thought you probably had never heard about. There's no neurons there. These are very highly secretory cuboidal epithelial cells. And we actually found that we could get choroid plexus to grow sometimes spontaneously in our brain organoids. And so then what Laura Pellegrini, who is a postdoc in my lab, she'll be starting her own lab down at King's in London. What she discovered was a way of getting our brain organoids to generate much more of this choroid plexus, so much so that we could generate pure choroid plexus organoids. And then when we do this, we end up with these tissues that look like this. This is a dish that's a 6 centimeter dish in diameter, just to give you a sense of how big these things are. So they make these really big fluid-filled sacs, and then you can just stick a syringe inside and collect the fluid and analyze it. And so when we've done that, we've been able to identify essentially that fluid that's being produced is CSF. So it has the same sort of protein signature that you would see in actual CSF. And so the beauty as well of these organoids is that the production of that CSF inside of those fluid-filled sacs is a process of both secretion of new proteins by those cells themselves, but also filtering across a very selective barrier. So this choroid plexus, this blood CSF barrier is just as selective as the blood-brain barrier, because the CSF has free access to the brain. It can flow through the brain parenchyma. So anything that needs to get into the CSF, it needs to have just as a selective barrier there as the more highly studied blood-brain barrier. So we wanted to test whether these organoids also have such a selective barrier. And so this is where then collaborating with a chemist, actually Claudia Bonfillo, who was also here at the LMB and has now started her own lab, really came in very useful. And so what we did with her was to look at whether we could see the selectivity that we know exists in vivo to L-DOPA versus dopamine. So we know that dopamine doesn't cross into the brain, but L-DOPA does. But these molecules are so similar. And they're both, of course, very small molecules. So all of the in vitro models so far established haven't been able to show this kind of selectivity. So we wanted to just sort of like really push the boundary and see if our organoids could show that selectivity. And so she did some NMR for us on these on the fluid that we would extract from these organoids that had been where we've added L-DOPA or dopamine into the media. And we could see really nicely that L-DOPA did cross into the fluid, but dopamine did not. And then we did this with a whole bunch of drugs and basically compared our in vitro measurements to actual in vivo measurements taken from humans. So these are measurements from CSF and serum, and actually measuring how much of a particular drug do you find in the CSF compared with the blood, and then what kind of measurements are we seeing in vitro, and how does that compare? And what we saw is a very, very nice correlation. But what like totally blew me away was actually the slope. The slope of 1, basically this model is not only highly predictive, but it's actually quantitatively predictive. So basically from our organoid data, we could predict how much of a particular drug, at least when it comes to the drugs we've tested, how much of that drug you would need to get in the blood in order to get a particular concentration into the CSF. And then of course, from the CSF, it can get into the brain. So this is a really cool, I think, exciting new direction that we're moving in terms of, being a useful model to hopefully develop new ways of getting into the brain. And then these, it turns out actually that these choroid plexus organoids also led us into the whole COVID realm, because it turns out that these choroid plexus organoids express large amounts of ACE2. So we looked in all of we've done a fair amount of single cell RNA-seq as well in organoids, and we've looked in all of our single cell RNA-seq, and we don't really find much ACE2 in neurons or astrocytes or really most of the typically studied brain cells. But in the choroid plexus, we found these particularly the very mature cells, the very, very highly secretory cells of the choroid plexus seem to be expressing large amounts of ACE2. And so then we collaborated with Anna Albeca-Moreau in Leo James's group. And here she is actually in the CL3 working with the SARS-CoV-2. And we actually put some of this virus on these organoids and found that indeed the choroid plexus organoids showed quite a high propensity towards infection. And actually, kind of scary, we see a real breakdown in the tight junctions between these cells. And in fact, some of the first experiments we did, we went to go and collect the samples afterwards, and the organoids had just completely dissolved. It was quite striking, really. So so in fact, we've done further experiments and really shown that the barrier seems to be becoming very leaky after infection with this virus. And so we think this probably has some importance for some of the neurological symptoms that occur in some of these patients. Yeah, so that's it for me. I just want to thank the lab as a whole. It's a really fantastic group of people to work with, but especially the collaborators that we've had in the LMB. We've had so many fantastic collaborations. And I don't know if you noticed, but I think every collaborator I just showed was a woman. So it's a really great environment to be in right now. And this is us in front of the cryos. I was so happy to have any excuse to do some cryo-EM. You're in LMB, you've got to do it. And of course, I want to thank my collaborators as well, and I'm happy to take some questions.
Thank you for the great talk. Are there any questions?
Beautiful talk, Madeline. Yeah, thank you so much. So I was just wondering to what extent you're using in vivo tissue versus in vitro comparisons. Mm-hmm. Kind of particularly where I was wondering was on the cryo-EM, for instance. Yes. So you showed those, how you can kind of go down to the subcellular resolution basically and so on. And is that something that you're studying in vitro or in vivo, or how do you think about the relationship between the two?
Well, so the cryo-EM kind of has to be done in vitro because you have to get thin enough in order to get vitreous ice. You can't do that on a piece of tissue. But we can compare with, it doesn't have a cryo-EM, we can compare with, standard plastic embedded EM. And so some of the things we find, for example, is very, very thin ER tubules that essentially have no lumen. and it's completely smooth. And so this is already telling us something about the primary role of ER in these axons is probably in lipid transfer, because it's incredibly complex. You have this incredibly complex surface area to volume sort of, and of course you think about an axon, the surface area to volume of an axon is really special compared with any other cell. So, and particularly growing axons, obviously they're going to need lots and lots of new influx of lipids to do that. So we're already learning something there, but classic EM it's hard to see that kind of resolution, right? So you wouldn't, and for example, you wouldn't be able to see that kind of single, a cargo on a vesicle like that. So that's tricky to do, I think, on an actual in vivo setting. So sort of combining these different techniques, I think, really gives us a special ability. What we can do though now is start to compare with other. So what we're going to be doing now is comparing with other species, actually. So look at, like, for example, mouse or, even like fly neurons. Can we see differences there that explain some of the differences that we know also exist on a cell biological level? But for example, like with single cell data or that kind of thing, we always like to come back to atlases like those you've been doing to really compare the data that we're generating with actual in vivo data. Yeah.